Distinguished guest, researcher, scientist, we are welcome you for the first session, technical session of this Kazakhstan, Pakistan, Turkey Youth Forum. We have this 5 presentation in this session, and the first presentation is by Professor Dr. Shahid Mansoor is here, and he will present the lecture on overview of agricultural biotechnology in Pakistan. So please welcome.
Assalamu alaikum. Bismillahirrahmanirrahim. I'm just waiting for the slides to be shown. Before I start this presentation, I would like to have some acknowledgement and thanks. First of all, is about Comstech. They have been very kind to invite us for this presentation, and also this is the third forum or meeting that we are holding. So the first meeting was in 2019, which was funded by ICDB and Comstech, and then then the next meeting was delayed by about 2 years because of the COVID, and then we thought about that whether to have it on hybrid mode, but ultimately we were successful. So it was held last year in V2 in Turkey, and now we are again back in 2022 in Pakistan. So before I start, and I must acknowledge also all the work that has been done by my colleagues and students, and also is the compilation of a lot of work which has been done by a lot of members. So first acknowledgement is to all those researchers that have contributed to this. So the first thing is that the world population is increasing at high rate, and hunger is killing more people than what we think as like more disease like AIDS, malaria, and tuberculosis. And then the elimination of hunger remains one of the major challenges even in 21st century. And then the zero hunger is the top agenda of sustainable development goal of United Nations. So I think that this is something which is very important, and I'm very pleased that our president has very nicely elaborated on some of those challenges and opportunities. Another very important challenge is climate change, which is an immediate issue. And if you look at this by World Economic Forum 2021 and 2022, that some of the most important challenges are climate action failure, extreme weather, and biodiversity loss. And then so you can imagine that this how important it is this, and the situation in Pakistan. So obviously it is very pronounced in Pakistan that a population growth rate is one of the highest in the world. Climate change and COVID and environmental degradation are major issues, and then COVID-19 epidemic, and recently that the climate change and floods. So these are all point out that we need to develop and use technology. But fortunately, there are major opportunities because of the understanding in genomics, genetics, and genetic engineering. And there are several major disruptive technologies that have been developed due to innovation in biotechnology. And 21st century definitely belong to biotechnology. So one of the questions that if you go to any planner, they will ask you that what are the possible options if you want to have enhanced food production. So one thing is that definitely is that we can have some intervention by management, like water reservoirs. We need water reservoirs, We need what rainwater harvesting is very obvious in recent flood and cropping pattern and livestock shift. This is something that is we require, and then the tree plantation is something which has been advocated by Pakistan. But the most in the history, if you look in the last 50 to 100 years, the major gain came from genetic gain, and so this gain is so important that with green revolution technologies, we were able to double the production of major grain like wheat within 5 years. So that means that genetic gain is not only in crop but also in livestock, meat, eggs. So these are some of the very important things is that we work on genetic gain. Obviously, somebody has to work on having better management of other issues. Okay, thank you. So there are two ways of developing or technologies which are available, what we call as conventional technologies, which was based on Mendelian genetics, and then also by using mutations. And we had green revolution, which was based on high yielding varieties. The second, this gene revolution has already, or green revolution has already leveled off, which means that we are not able to have further increase in productivity. So what we need is another revolution, which we call as gene revolution, where you can start with something like simple like tissue culture, then genetic engineering and marker-assisted selection. And what we advocate is that we have unlimited gene pool, because the breeders depend on the gene pool for the further improvement. So biotechnology, obviously, when we do genetic engineering or genetic manipulation, we need biosafety rules. So there are policy document that tells us that we have all the possible policy document that, and we have listened to the president of Pakistan as well, that this is something which is very much part of our planning, like 5-year plan by Planning Commission, medium-term development or Vision 2025, and science, technology, policy, innovation. So these all documents are available, and also there are some document done by PCST and Pakistan Academy of Sciences. So also we have regulatory system, because we need some laws. So the first law was Pakistan Patent Ordinance, then establishment of biosafety rules. It took a lot of work and hard, it was a hard nut to crack, and then plant breeders' right, and for medical related activities, it is controlled by Drug Regulatory Authority. So what we can see from this slide is that our regulatory system is all over the place. There are several ministries who are controlling this. So this is something that it caused a lot of delays, because there's no coordination among different ministries. But at least we have the relevant law. So first example that I will give you is a success story that has been developed by NIPT through all my colleagues and students, where we were able to develop transgenic cotton with 3 genes all together. It was a big challenge, because putting all the genes together and then transforming and then getting expression was something very interesting. And luckily we got very good expression in all leaves, bole rings, and immature seed of NIPT in triple gene cotton, which means that they have got 3 genes, 2 for insect control and 1 for herbicide tolerance. And these genes are now in the field, and I'm very happy to share with you that they have, for the last 2 years, they have shown consistent results in field trial, and we are hoping to take these lines into field trials next year. So now another example is that we discuss about the problem of climate change. So we need genes that can protect us from drought and salt, which are major stress. Heat is another stress. So this is an example where we learn from plant, and we identified 3 genes that are isolated from plant themselves. And what we are doing is that we are putting them together in one construct, and we transformed into cotton and canola. The reason is that cotton is the only crop which is genetically modified crop, which is commercialized in Pakistan. And by working together, by linking again 3 genes together, we were able to produce plants that are both cotton and canola, and those plants have been tested under glasshouse condition, and plants show drought and salt tolerance. But the more important thing is that there was no yield penalty, because some of the material which has been commercialized in other parts of the world for drought tolerance, they have a yield penalty. Now there are challenges whenever you talk about GM, there are some there's always 2 groups of people, some who agree with the GMO, and there are others who don't agree. Although that we have to this is a very important decision that nations have to make, that what is important. So I was talking to a Chinese professor, and he said that for us the most important thing is to feed the hungry mouth. So what we can do is that we can either educate our policymaker, or we can adopt new technologies. Fortunately, there are some disruptive major technologies which have emerged in the last few years. These include genomics, genome editing, gene drive, synthetic biology, and then nanobiotechnology and big data science, bioinformatics, artificial intelligence, and precision agriculture, which are going to have or already have major impact on agriculture, health, industry, environment, and also there are also some threats of some of those technologies. So what is so important about genomics is that the speed and the cost reduction in determining the whole genome sequence. So remember that how much was the cost of the first human genome when it was sequenced, and now we are talking about a few $100s to sequence a human genome. And this is something that it is going to have an impact in breeding. It is there, so I'll give you a few examples. And one example that I can give you is the genomic selection in livestock. It is happening both in plants and livestock, but livestock, somehow it has already created an impact for the last 15 or 20 years. It has been practiced, say that by using genomics, you can predict the accurate genomic or estimated genomic value of the bull or female cows. We can also have breed specific markers, and also so what we are trying to do is that we establish this technology, and then we are going for the customized DNA chip or SMP chip. And our major focus is on the dairy selection, and also buffalo, because more than 50% of milk in Pakistan comes from buffalo, because we love buffalo milk. Is very thick. So to start this work, again, this was something that was done by our students. So we have now whole genome sequencing of all cattle and buffalo breed of Pakistan, and services for genomic selection. We are already providing services, and customized chip is designed, and then sex human is another to have a fast track. So I hope that whenever we meet next time, we should have a session on genomic selection in livestock. So the other thing is that obviously everybody knows about this. So I may not have to tell a little more about this. So that we can also use genome editing, where we can bring very precise change in the genome by using CRISPR technology, and the specificity of this system is based on guide RNA. So guide RNA tells where to make this, and this is perfect because we can do multiple genome editing at the same time. So there is a lot of recent development. So we are following this. So probably those who follow publication from our group, they know that we are looking at the development in these tools. So for example, how much, what are the different types of Cas variant which are available, So the idea is that you make the size of the Cas protein smaller, so that it can be packaged into viral genome, or even it is easy to deliver whenever, if you are using other than bacterium technology for the delivery of this. So last time when we met in Turkey, there was a very interesting talk by a speaker from US. I mean, she was actually a Turkish of origin, and there was a talk of using this carbon nanotube for transformation. So what we need is a precise and smaller size Cas protein, and there's a lot of variations. So I won't go into the detail because of the time. So I'll just give you some of the example of genome editing. So obviously, there's a lot of work to enhance yield. Now, this is something that was unexpected. This is for the first time that we are able to address the issue of yield increase, because we precisely know what are the genes that are contributing to the yield of the plant. And so by targeting those genes, so earlier in genetic engineering, we were mostly talking about controlling disease and pests. So this is an example of where we are targeting negative regulators, and this is the first step. That this is something very easy. Anybody who is working on CRISPR, they know that if you want to target negative regulators of yield, so there are 5 genes that we have targeted in CRISPR, and now we have the phenotype, and inshallah soon we will have a plant so that we can compare the yield of the plant. Another very important thing is about the genome editing in wheat, because something which is right, starting from Pakistan, Afghanistan, Turkey to Central Asia, we all love bread, and we need to have more yield. So for this purpose, we are editing again multiple genes which are related to yield increase. So increase the grain size, increase the number of tillers, and this is something that these are all factors that contribute to the yield. And we already have some plants, so hopefully in coming months we should be able to. And then our favorite topic is obviously to create disease resistance. And so one of the things that we were able to consider about 5-6 years back is that so far we have been focusing on disease resistance genes. This is the first time that we started to think about susceptibility genes. What is the most important component which is required by the pathogen to enter into the cell, and we were able to create virus resistance by targeting some of those susceptibility genes, and then create basmati rice resistance to bacterial blight, because these are sweet genes which are sweet genes which are required by the pathogen. So if you target all 3 of them, theoretically you have immunity against this bacterial blight. So by the way, this is all published. The next very important revolution which is coming in biotechnology is the gene drive. Now, this is the first time that we are able to quickly deploy this transgene into the plant. So this is something that very important. And earlier it was developed for insect, and now it has already been developed for plants. Another very important area is synthetic biology, where we can develop synthetic genes, and this is very important for. May not be very important for the crop plants, but there are a lot of products that we use which are made in fermenters. So synthetic biology is very important area where a lot of those microbes can be engineered so that they produce a product at a very high efficiency. Speed breeding, I think that Dr. Nasir is going to speak about this, that we can fast track breeding process, because it takes about something from 4 to 10 years to breed a variety. But by using speed breeding, we can reduce that time cycle. Another very important area which the president also mentioned is the improving photosynthesis. I thought that probably he has read my. So this is something which we always think that there are two issues that can be addressed if we can enhance the photosynthetic ability. One is that in photosynthesis you are consuming carbon dioxide, which is a major greenhouse gas. And improving photosynthetic efficiency can enhance food production and mitigate climate change impact. And there are, for those who work, I won't go into the details, but there are, for scientists who work on and think about this, there are very important intervention area, like we can engineer synthetic CO2 fixation pathway, or engineer synthetic photorespiration pathway, or introducing CO2 concentrating mechanism, or improving the catalytic activity. So these are some of the very important areas, because it is going to have a huge impact on the crop productivity. I will go another area, which is biofertilizer.
4 minutes.
So that's an area that because fortunately there are some, as a collective conscious, we are saying that in the last 100 years we have done a lot of damage to our environment by using a lot of chemicals, especially the pesticide, herbicides, and then also fertilizers. So at least if we can use less fertilizers, or at least by 25 to 30%, or maybe so what we have done is that we have set a target of 25 reduction by 2025, and I'm very happy to tell you that we are getting some very interesting data, and I hope that Dr. Asma Imran will have some discussion on this. This is an area where there are two advantages of using this biofertilizer. One is that obviously to reduce the environmental impact of the agriculture, and also I mean, another intervention could be that if these biofertilizers are combined with organic matter, then obviously it's going to have a lot in improving the soil of health of soil. Then come the big data science, artificial intelligence, nanobiotechnology, and precision agriculture. So obviously on top of these management issues, we used to depend on manual labor, but now we have these tools which are available. So for example, if you want to have an overview, you can use satellite data, and if you want, want to have a data on the actual crop situation, you can use drones. And by using this technology, there was some experiment done, and the precision of the data is about 95% accurate. So this is something where that so what I mean to say is that not only we have to work on the biology, but also work in collaboration of scientists like space scientists, or those who work in artificial intelligence for precision agriculture. So the way forward is that we need climate smart agriculture. This is climate change is there. We had already experienced this, all the situations that we are seeing this, but believe me that having a lot of water, if you can conserve that water, this will be going to be a blessing. We need a technology platform for crop improvement, technology platform for livestock improvement, because it's a very important component of our overall. Another very interesting area is the microbiome. So we all have our microbiome on our surface, in our gut. So it is already there. So this is a mind-boggling data which is coming, and then enhancing soil health, because healthy crop will come only if we have healthy soil. And last but not least, and I'm very pleased that I see faces which are very familiar, and I see friends from Turkey and other places, And I hope that we will have the similar kind of intimacy with our friend from Kazakhstan to have international linkages through OIC platform. There are some very important initiatives which have been taken by comstech. And with these words, I thank you very much.
Thank you, Raksa.
So we have question session answer later on. Thank you very much for your kind presentation.
So our second speaker of this technical session is Dr. Emre Aksoy. He will deliver a lecture on plant genetics and genomics for building sustainable agriculture.
Hello, everyone. My name is Emre Aksoy. I am from Middle East Technical University. First of all, I would like to thank all the organizers, Comstech, to invite us to give this interesting seminar on biotechnology, especially agricultural biotechnology, to basically facilitate youth development in OIC countries. So today, I would like to talk about the importance of the plant genetics and genomics for development of sustainable agriculture. As we all know, the population is increasing in the world, and it is expected to reach to almost 10 billion by 2050. And of course, this is causing a large food security problem, which basically indicates that until this time, we actually need to increase our food production by 70%. And actually in the past, we had succeeded with the help of the Green Revolution. But at the end, what happened was luckily in major crops, we increased the yield and production. Unfortunately, while we have been doing this, we contaminated the soil and the water systems because we used a lot of fertilizers. So here the graph is representing that on the left side, the amount of the nitrogen, potassium, and phosphorus is increasing, especially the nitrogen fertilization. But unfortunately, it contaminated the important rivers of the world. And this at the end led to the contamination of the important water sources, including the Gulf of Mexico, which led to the, some oxygen deprivation in the water, unfortunately leading a mass important loss of the fisheries. And of course, we are aware that the global warming is triggering a climate change. And unfortunately, we have been struggling with this in the last couple of years in different parts of the world. We already know that the temperatures have increased almost 1.5 degrees, and we expect that it will increase furthermore. According to two different models, the first model is actually representing that the increase will be around 3 degrees. The other one actually expects it can reach to 5 degrees at the end of the century. So this will be a huge devastating result. And this increase in the temperatures actually doesn't happen everywhere similarly. What we observe that in some parts of the countries, there will be some anomalies in the temperature. In some parts, for instance, this image is representing in the Northern America that the temperatures are dropping. Unfortunately, in the Russian section of the world, it is increasing. So these anomalies will actually cause a lot of production problems. And these images are representing what type of effects the climate change can actually result in. So we will see a lot of floods in some parts of the world. On the other hand, in the other parts, we will see more droughts. And unfortunately, it will also cause salination problems in the soil. And of course, we can expect that there will be more biotic stresses on crop production. Now, actually, seeing all these effects of the climate change, United Nations actually came up with some important sustainable development goals and targets. And genetics, plant genetics and genomics can actually help us to reach to some of these goals by 2050. So before showing you some examples toward these goals, I just want to emphasize the importance and how we can utilize the genetics and genomics in the plant technology. So in the past, we actually have developed major crop varieties by screening them from the wild relatives. After that, we domesticated them, and finally, we developed the genetically assisted selection or little alleles in the varieties. But by doing this, basically, we narrowed down the genetic diversity in the crops. On the other hand, we have some neglected crops already available, which represents a larger genetic pool of the wild relatives. And since we haven't done their domestication yet, they are available highly. On the other hand, what we need is actually we need to connect or combine the advances that we learned from the development of the major crop together with the neglected crops and combine them with the fundamental research to determine a loop between the bench, field, and fork. And actually, while we are doing this, we need to use the orphan crops. So here you can see on the left side that there are the major crops that actually are stored or protected either in the gene banks or on the farm or in the wild. However, we keep majority of the seeds, especially in the gene banks. On the other hand, if we just look at the new developed crops, then we actually keep them in the wild. So we need to do more research on those aspects. On the other hand, the orphan crops, we can utilize the technologies of the major and new crops. And we can actually utilize different types of breeding technologies to develop them much efficiently. And more interestingly, we can use already the available information in the crops, which actually look very similar to the background of the orphan crops. So we don't need to repeat all those experimentation in the orphan crops. We can use the high genetic diversity of the orphan crops. Here, for instance, as an example, if you just want to deal with the amaranth, then you can actually take the technology developed in the soybean and just integrate easily in the amaranth and facilitate its breeding quickly. So this translational genomics actually will dominate the agricultural research in a way that we already have the genetic resources, we can use them, we can actually use these resources together with the genomic technologies. For instance, we can just use the reference genomes, we can use the pan genomes, we can do resequencing, transcriptomics, and other omic technologies altogether. We can determine some genetics, some candidate genes, develop markers, and we can incorporate all of them into the applications of the genomics. So on the right-hand side, you can see that there are many different applications of them. For instance, we can use these markers in the marker-assisted selection and backcrossing, or we can use the genomic selection, etc. And of course, while we are doing the genomics, we will produce a huge, massive amount of data, and we need to be able to analyze them. In the past, of course, we have developed different platforms to understand these data. However, now the amount of the data is even larger, and it can actually make confusions. So what we need is a good system to integrate these different data and actually choose the best known information out of it. And to do this, now we need to get the next generation of the artificial intelligence. And nowadays, there are many different groups in different countries working on integration of the artificial intelligence in the analysis of this genomic data. And then, of course, while we are doing all these, we need to make sure that we are always keeping the genetic diversity in the crop improvement. So in the nature, we have the crop wild relatives. We have developed the cultivated crops we have the land races, etc. We have been actually doing the genomics, the sequencing of the crops all the time. But now we need to go back to the large genetic pool to actually look at the differences between different crop wild relatives and understand what type of genes are actually behind these differences. So if we can identify these variations, sequence variations, then we can understand what type of traits can be elaborated with the help of these newly identified genes and convert them into markers. This graph, this map is actually representing you why we need to conserve the genetic diversity. As you can see in some parts of the world, the diversity is so large and we need to protect it and use it for future developments of the crops. Here it is, as you can see, Turkey is like the hub of the genetic diversity for many of the different crop species. Now I will show you a few examples that have been already done that uses the genomics and plant genetics in order to actually lead to the UN targets. So this image on the right is representing a field trial from Ethiopia, where actually they have used a diverse durum wheat genotypes, and they actually determined which genotypes are drought tolerant. And among these, they found that one of them actually yielded 61% better than the rest of them. And we have been doing a very similar experiment in our greenhouse under hydroponic conditions. We have been phenotyping the roots of a large genotype pool of the durum wheat, and we identified some of them are really tolerating the iron deficiency very well. So the main idea is actually trying to bring them into the field and to spend them or distribute the seeds to the farmers. And meanwhile, we have also their genetic sequences. So with the matching of the genotyping and phenotyping, we are also determining some specific markers that can be used in future breeding programs. Another one is actually a very good success story coming from Harvest Plus. So in Harvest Plus, they have actually looked at to the amount of the iron in the seeds of different bean varieties, and they found that 10 of them are really successful. So they contained more than 2-fold of higher iron in the seeds. So what they did, actually, they took these seeds, they distributed the seeds in different nations in Africa. Among them, there was Rwanda. And then the farmers, they took the seeds, they grew them successfully. And then, of course, they started to consume these seeds. And later on, they actually took the blood samples after just a few months of feeding, consuming these seeds. And luckily, in Rwanda, the amount of the iron in the blood increased 55% just in a few months. That was amazing, a huge success. So just by looking to the seeds, screening them and finding the best genotypes, that's it. We just can use them, utilize them. And on the right-hand side, that packet, the product now is available in the market. This image is coming again from Africa, South Africa. They again use the genetic diversity of different crops. The image on the right-hand side is representing actually a cassava experiment. So they did a drought experiment, and they identified the ones that can tolerate it more and adapt it for South African climate. So in the last 2 slides, I would like to show you how actually genetic experiments can lead to gender equality in different countries. So we have a project right now together with some countries One of them is Jordan. We are focusing on the development of some vocational skills in Bedouin women. So this basically this population is some part of it actually still lives in the urban areas of the Jordan, and unfortunately they are poor, and especially women are undereducated. So in this project, supported by the European Union, we plan to go there, train them how to produce the crop products that are adapted to Jordan climate. So with that, we will teach them how to make the business models and develop their own crops and sell them. And another project recently we submitted to the European Union. In this one, now we are actually planning to develop new food products from the different pulses that are adapted into 4 different countries: Portugal, Spain, Turkey, and Egypt. We will basically first find the best genotypes or varieties that are highly adapted and nutritious, and then we will use them, combine them to develop new food products like crackers or something. And then we will develop a business model with that. And at the same time, we will distribute them to the different children in the high schools, and we will look how much the protein and nutrition levels increases in their bloods. So as you can see, plant genetics and genomics not necessarily only increases our production and facilitates our understanding of the breeding, but at the same time, it can lead us to teach and train different parts of the population. With that, I would like to thank everyone, and I think at the end of the session, we will take the questions.
Yes, definitely.
Thank you.
Thank you, Emre. Thank you, Dr. Emre, for your nice presentation. We have next presentation on plant biotechnology, past, present, and future by Professor Dr. Leyla Açık from Department of Biology, Gazi University, Ankara, Turkey. Please welcome Dr. Leyla Açık. Much better working. We have generally 15 minutes for the presentation. Okay. Thank you very much.
You couldn't manage in the first one.
Maybe some type of.
Hello, everyone. I am from Turkey and I work at Gazi University, not Ankara University.
Sorry.
No problem. It is great pleasure to be here. Everyone is talking about now and the future biotechnology. I will take you to the beginning of the biotechnology. I will talk about biotechnology and first generations techniques. And I won't talk about recent techniques. I will just mention it. And I will talk about what's happening in GMO in Pakistan, Turkey, and Kazakhstan. And I will talk about a bit advantages, risk, and concern. What do insulin, COVID vaccine, antibiotics, and stonewash genes have in common with golden rice, cheese, bread, and beer? Of course, they are all products of modern biotechnology. Biotechnology starts with civilization. Since beginning of the civilization, while our ancestors still able to influence DNA and other organisms. In that time process called selective breeding, artificial selection, without they know biotechnology, they used to use yeast, make breads, and fermentation for cheese making and others. And first, biotechnology term used by Hungarian engineer in 1999, and he described as goods to be made from raw materials using living organisms. So what is biotechnology? Biotechnology can be broadly defined as using organisms or their products to modify or improve human health and human environment. There are lots of types of biotechnology, and the most important part is genetic engineering. Genetic engineering is the process of transferring genes between organisms or modifying genes in the organism to remove or edit desired trait or characteristic. If you look at recombinant DNA technology, genetic material from one organism is artificially introduced into the genome of another organisms. I was planning to talk about two important projects on biotechnology, but I will skip one and I will just concentrate into insulin project. In 1972, some researchers, including Boyer, realized that the enzyme EcoR1 cut the DNA in such a way that the ends were not blunt and cohesive. Boyer and Helling began their effort to create rDNA to insert into bacteria by trying to cut DNA with EcoR1. And they did lots of works, but they haven't succeeded first. Another scientist, Cohen, in lab discovered DNA different than chromosomal DNA, plasmid DNA. He is working on plasmid DNA. And Boyer and Cohen plan collaborative project to discover what genes are present on plasmid and how they are arranged. So they join tetracycline resistant plasmid with calamicin resistant plasmid, and they insert into E. coli, and they show that genetic materials could indeed be transferred between species. They did some work with Staphylococcus as well. Then Genetics, first biotechnology firm founded in 1976 by Boyer and Swanson. So if we look at the insulin project, in 1921 and 1922, Canadian teams discovered insulin, and they making insulin for the treatment of diabetes. But in that time, they get insulin from living organisms, especially pigs and cows' pancreas glands. According to InDiabet's forecasts in that time, more than 2 tons of pig parts were needed to extract just 8 ounces of purified insulin. Can you imagine that? Their initial goal, the scientists of this breakthrough in diabetes therapy, were directed towards improving chemical DNA synthesis. Itakura, another scientist, was doing chemical DNA synthesis. Riggs, Boyer, Cohen, and Itakura get together and they wrote a project, insulin project. I will just skip this. They applied to get grant, but grant was reviewed moderately well, but not founded in that time. 1977, Boyer and Itakura succeed in expressing a mammalian protein, somatostatin, in bacteria. Their first game was insulin, but somatostatin is smaller, so they did it with somatostatin, and they got the money from rich friends. 1978, Boyer and Itakura constructed a plasmid, and they cloned human insulin. That's the team. in case of recombinant insulin, Eli Lilly, another company, signed a joint venture agreement with Genentech to develop production of process for Humulin. In 1982, Humulin was approved by FDA and it became the first biotechnology product to appear on the market. It was the first drug produced through recombinant DNA technology. So it's, you can see the history. If we look at a bit plant biotechnology, farmers have been using traditional modification methods like selective breeding and crossbreeding to breed plants with more desirable traits for thousands of years. The recombinant DNA technology have allowed humans to more directly and quickly alter plant genetics. I was supposed to talk to students, so I start basic things like tissue culture. It's very important to do tissue culture first. And then you need restriction enzymes to cut the DNA. And plasmid, you need to move DNA from one place to another place. And you need marker genes to select what you put into organisms, and reporter genes, selectable marker genes, had the surgeons select transgenic plants. You need strong promoters, and then you did cloning and then select the plant. First generation techniques, if you look at it, like you use bacteria, Agrobacterium tumefaciens, and you can do electroporation and PEG-mediated delivery, protoplasm fission, microinjection, and biolistic and nanoparticle-based gene delivery methods. It's amazing bacteria, Agrobacterium tumefaciens. Normally, it's cause Crohn gall disease in many plants, but it's like a genetic engineer now. This bacterium has a big plasmid in it, and has tumor-inducing genes, along with additional genes that had tDNA integrate into host chromosome. It's going out from bacteria and go to plants. And scientists remove some part of the tDNA and integrate foreign gene into a genome of cultural plants. In electroporation method, you give strong electric fields, pulses, that alter cell permeability and generate some pores, and then you give the DNA, and then cell repair themselves. Another technique is gene gun, and you cover the DNA with gold or tungsten, and then give the DNA into the plant. One of the first experiments in plants was try to find, instead of pesticides, give insecticide resistant, instead of insecticides, you give insect resistant genes. There is, again, bacteria, this time is Bacillus thuringiensis, is gram-positive facultative aerobic soil bacterium. It's synthesized crystalline inclusion containing protein known as delta endotoxins, or cryproteins in short. These proteins have insecticidal property against diphtheria or Lepidopteria larvae. It's used in the first against many agricultural insects such as Lepidopterian, Coleopterian larvae. 1987, Plant Genetics System was the first company to genetically engineer insect-resistant plants by incorporating genes that produce insecticidal proteins from BT into tobacco. This is another interesting story, but I'm not going into that. Crops bioengineer food pest resistance could increase yields, eliminate the use of several insecticides, now derived from fossil fuel, and reduce health risk and groundwater contamination from pesticide. another important things with plant to get virus resistance, to insert gene from viral protein into plant cells via agrobacterium. So many people were trying to do this, and they work with different promoters and they couldn't get a good result. And then in the end, 35S promoter helped them to get good yields. And this time, 1980 and 1981, scientists considered the possibility of developing transgenic plants that would express sequence derived from tobacco mosaic virus. This is very important project, and Powell and some other scientists together, they managed to clone CP gene into the plants. And in the end, they get resistance to infection by TMV. First genetically engineered crop plant was tobacco. It was developed creating a chimeric gene that joined antibiotic resistant gene to the T1 plasmid from agrobacterium. And tobacco was infected with agrobacterium to transform with this plasmid, resulting chimeric gene being inserted into plant. Through tissue culture techniques, single tobacco cell was selected that contained the gene and new plant grown from it. Finally, transgenic virus-resistant plants were first produced in 1986. China was the first country to commercialize transgenic plant, introducing a virus-resistant tobacco in 1992. And then studies tolerance to salinity, osmotic stress, resistant to herbicide. There are so many. And the other work was on to improve the quality of the plant. For example, improve storage in tomato with antisense technology, transgenic flower color and shape, and transgenic for nutritional quality. Now with this technology, you can transfer gene not from one species to other, from one kingdom to another. And golden rice is famous with nutritional quality works. Now plants use the bioreactor to get, for example, PHB or vaccine and other peptides. Why do we use plants? Because renewable, easy, scalable, cheap, global, diverse, and especially new technology, we can do lots of things, especially with CRISPR-Cas technologies. And I know there are so many experts here, so I am not going to talk about that. This is very interesting, horizontal gene transfer. People used to do this one in the past. Farmers used to do this without knowing what are they doing, but that's good transgenic making. I'm not going to talk about these genome editing technologies because I saw CRISPR-Cas is going to talk by others. I just wonder what is happening in Pakistan, Turkey, and Kazakhstan with GMO. I am surprised that Pakistan is good at this. Not surprised, I'm happy about that because we don't have anything. But Pakistan, cotton is the only crop under commercial production in Pakistan. Pakistan did some GMO. Turkey are no GMO plants under development or commercial purpose in Turkey because it's forbidden. Kazakhstan, the same things. Since the 2015 amendment to seeds law, which prevents full field trials or commercial production of genetic engineering events, research and development in Kazakhstan has been constrained and testing limited to laboratory greenhouses. Why they this kind of rules? Maybe they afraid of their risks, but there are lots of advantages as well. Foreign genes can be introduced potentially faster than traditional plant breeding. Specific gene can be transferred much more controlled than traditional plant breeding. And you are doing this for benefit human health. There are risks. Molecular characterization may be difficult. Food fit risk assessment, environmental risk assessment, I can count this. The other things, herbicide resistance or insect resistant genes could be spread from the engineered crops to wild relatives and create weeds that are especially difficult to control. Some scientists fear that USDA doesn't require sufficient precaution to prevent the spread of genes, engineered plants to their wild relatives. But CRISPR-Cas, maybe we won't have this kind of problem. As a last words, global agricultural production is facing as many challenges. By 2050, the world population will reach 96 billion and the demand for main crops will have increased by 60%. Detrimental climate change is affecting and going to affect plant production. Income level will be multiple of what they are now. In order to feed this larger, more urban and richer population, food production must increase by 70%. Annual cereal production will need to rise to about 3 billion tons from 2 billion tons. Annual meat production will need to rise by over 200 million tons to reach 470 million tons. The global food crisis of 2008 reminds us of the importance of innovation in agriculture to address global challenges such as population growth and climate change. Additionally, Russia's war with Ukraine is preventing grain from leaving but now it's okay, that problem, but it will be another problem next. Additionally, farmers will have to hit targets for reducing greenhouse gas emissions, improving water usage efficiency, and meeting the demands of consumers for healthful food and high-value ingredients. In this context, new plant breeding techniques are needed to contribute improvements in crop productivity and sustainability.
Ma'am, we are 5 minutes over.
Although biotechnology has potential risk, starvation is worse. We should be shameful. People in Yemen is dying from hunger. Thank you.
Oh, sorry for that.
Okay, our next
Thank you.
lecture is on genome editing for crop improvement and alternate delivery methods by Dr. Imran.
Thank you very much, ma'am. If you see the schedule, by now I should have completed my talk and say thanks to you. My actual plan was to give you some practical examples of genome editing, which we are doing in our lab. I'll be very quick in case of introduction part. If you see the CRISPR-Cas system is basically the natural infection, is the natural response of bacteria against invading viruses, where they acquire some part of the DNA of that bacteria. And then it is incorporated in the CRISPR array, and CRISPR array worked along with Cas9 protein, and that this particular guide RNA, it works with Cas9 and it cleaves the DNA or the RNA depending on the Cas protein, which has its homology. How we do this in crop plant is that we have Cas9, we develop a vector, we have Cas9, which is transcribed under a promoter, and then we designed our guide RNA, which are here. This guide RNA is basically the target of the DNA where we want to make the changes or in the genome. And then it is also transcribed under a different promoter. We develop a cassette, we do the transformation, and then this Cas9 is transcribed and translated, and then the guide RNA as well, and they work together and they target the gene. Cells usually tend to repair themselves. So whenever it's cleave the DNA, cell try to repair itself, and there are two mechanisms which are involved, which is one is non-homologous end joining, where you get the indels, and other is basically the homology dependent repair. Now we basically exploit this procedure or this process, natural process, in case of CRISPR-Cas, and we can do targeted gene mutation, we can do the knockouts, we can do the knock-ins, we can work on synthetic biology, we can work on gene therapy, and we can also work on gene regulation by various kind of defective Cas proteins. I'll tell you a few of the projects which we are doing in our lab. The first thing, we are basically a virology lab, and our first aim was to develop genetically modified resistance against viruses. And the viruses in which we are interested are basically the Gemini viruses. These are single-stranded circular viruses. There are 14 genera. And the most important are bigomoviruses. This is important for us because they infect cotton. And cotton is, there is a disease which is called as cotton leaf curl disease. And Pakistan is, according to an estimate, suffer about $2 billion per year because of this disease. And $2 billion for Pakistan is a huge amount. It is transmitted by whitefly, and there are two kinds of, basically three kinds of viruses, which the bigomoviruses. One is monopartite, having a single genome, bipartite, having two genome, and then monopartite having some satellite molecules. The cotton virus is basically a virus that has bigomovirus and some satellite, which we call beta satellite and alpha satellite. So what we did was that we targeted cotton leaf curl Cochrane virus Burewella strain by genome editing. We design guide RNA on that. The guide RNA which we designed, this is basically, we call it intergenic region. This intergenic region is the promoter region of the virus. We designed guide RNA of that. These guide RNA then were cloned in various, in the vector. The vectors which we used were developed in our lab. They were where the Cas9 was transcribed under 35S promoter, while the guide RNA were transcribed under U6 promoter. We transformed these cassettes into a model plant, which is Nicotiana benthamana. We developed transgenic plants, and then we challenge those transgenic plants with the virus. We have viral clones in the lab, and we challenge and we study how they are behaving. This gel photograph shows you basically the conformation of the genes, and these are then infectivity assay. This is Nicotiana benthamana model system. These were the non-transgenic wild type plants which were not infected. These were the wild type Nicotiana benthamana which were infected with cotton leaf curl Cochrane virus Burewella strain. And if you see the symptoms, they are very much like in the cotton, downward curling, stunting in the growth. And if you see these transgenic plant, we were unable to study any symptoms or observe any symptoms in these plants. We confirmed these plants with by doing southern analysis, where cotton leaf curl Cochrane virus Burewella strain was used as a probe. And if you see, this is a PCR positive. This one is the non-transgenic wild type plant which was not infected. These two in lane 3 and lane 10 were the plants which were wild type but infected with cotton leaf curl disease. And you can see the presence of a very strong band, but we were unable to see any virus which can replicate in these plants. Then we did some PCR analysis. The size of this virus is 2.8 kb. And if you see, this is the positive control of 2.8 kb. We were unable to detect any virus or any virus of 2.8 kb. However, we were able to observe some breakdown products or smaller products of of viral origin, which means that our CRISPR construct is basically functioning and it is not allowing the virus to replicate, at least in Nigritiana benthamiana. We also confirmed it by sequencing, and we have found that none of the viral particle having an intact intragenic region. This was a region which was targeted by CRISPR-Cas. So in conclusion, we were able to develop resistance in a model system using CRISPR-Cas by targeting intergenic region. Another project is again on cotton, which we are working. This is basically the reduction of toxic gossypol content in cotton seed via CRISPR-Cas under seed-specific promoter. Those who are from having a little bit knowledge of this cotton, gossypol is basically required in the cotton for its resistance against insects. But this gossypol is present in high amounts in the cotton seed, and that is why we cannot use cotton seed oil. It is not suitable for human consumption or sometimes for the animal consumption. So the challenge here is that you need a cotton plant that has gossypol in the vegetative part, like in leaves, but it should not have gossypol in the cotton seed. So here we explored the possibility of using CRISPR-Cas9 technology. And what we did was that in this particular project, we express Cas protein under a seed-specific promoter, which is alpha globulin B gene. While the guide RNA, they were under U6 promoter. So the hypothesis was that in the seed, only Cas9 will be produced in the seed, but Cas9 will not be produced in the vegetative part. So we designed guide RNA. This is basically, this PCR shows you the amplification of the alpha globulin gene from the cotton. This shows you the vector we had. In that vector, it was 35S promoter, which was transcribing the Cas9. So we removed the 35S promoter, we incorporate our promoter in that particular vector, and then we clone our guide RNA, and we confirm the guide RNA by the PCRs. Sorry. So this is basically the final vector where the Cas protein was expressed under alpha globulin B promoter, while the the guide RNA were expressed under U6 promoter. We did cotton transformation. We have a group, Dr. Shaheen is here. She's basically working on cotton transformation. We have had basically 3 constructs. The construct 1, this is the summary of that transformation. In one event, which is basically this, we were able to get normal embryo, while we were able to get some, embryos, but they were not normal, they were abnormal. This is the summary of the second construct, which was transformed. We were unable to get any embryo in that case. And this is the summary of the third construct. Again, we were unable to get normal embryo. So essentially, we got one event where we got one normal embryo. We use that. Here, if you see that this is the embryogenesis, then the shoot development, and then the root development, we transfer that plant, transgenic plant, into the soil, and then into the pots. And then this is the stage where the transgenic plant, it gives you the seeds, which we basically interested in. We confirm the transgenic plants by the PCR. This is basically the confirmation of the Cas9, and this is the confirmation of the guide RNA that it is expressing. It was basically an RT-PCR. We confirm the guide RNA, expression of the guide RNA at each step. When we got the seed, keep it in mind, we have the 5 plants, and all these experiments were done on on the first plant. This project is still going on. This is a HPLC analysis of the gossypol. This is the standard which we use. It was around 10 milligram per liter. And these 2 are from the transgenic plant. In this case, this is from the vegetative part, on the leaf part, where we would like to have gossypol remain there. And we were able to get 6.17 milligram per liter of the gossypol. And these are the results of the HPLC from the cotton seed. And in cotton seed, we got gossypol 0.8 milligram per liter. And usually gossypol is present in higher amount in the seed as compared to the vegetative part. So we were successfully able to reduce the gossypol contents in the seed part in this project. This is the project probably Dr. Mansoor has already mentioned. It was about the potato. We use CRISPR-Cas technology to control potato virus Y, which is a very threatening virus in Pakistan. So what we did was that we target the elongation factor in this case. This is elongation factor 4. In the same project, we also target the vehicular invertase to reduce the sweetening, cold storage induced sweetening. But here I'm just presenting the results of the virus part. We designed the guide RNA by targeting that gene. It was exactly like same. The Cas9 was under 35S promoter and the guide RNA were under U6 promoter. We did the transformation. We developed transgenic plants. And then we tested those transgenic plants in the small scale field by challenging them with the PVY. These are the phenotypes of the plants of the various transgenic line, and this is the DAS ELISA to calculate the amount of the virus. We studied those, we determined those virus after 7 days, 15 days, 30 days, and 16 days. And there were two lines, line number 16 and 46. They performed really well, and they showed the reduction in the PVY infection. We did some molecular analysis to see whether Cas9 is expressing there. We also study, if you see this is the expression of the Cas9. We did quantitative real-time PCR. This is basically the expression of the ELF4 gene. If Cas9 is working and your genome editing is working, the amount or the expression of the ELF4 gene should be reduced. And there were two lines where there was a reduction in the ELF4 gene. And when we study the virus in these two lines, the virus was also, because this is one of the, you may say, the susceptibility gene. It gives, upon expression, it gives more infection. And upon reduction, it gives less infection. So these two lines of potato, they shows very good resistance against PVY. In this project, we also use Cas13, which is another variant of Cas proteins. That Cas13 is basically act on RNA viruses. So we target this PVY using Cas13, and we were able to get resistance against PVY. Another area where we are working are the transient expression, because if you do the transformation, it is a conventional transgenic plant. There are certain methods which in the field, like RNPs, where you get your transformation, you get your mutation, but without the insertion of the Cas9. So one of the way is, as Dr. Mansoor has mentioned in his talk, is to transiently express the Cas9 protein. And for transient expression, viral vectors are really good. They give you very high expression, and they don't integrate into the genome. So what we did is that we are using potato virus X, and which we call it PVX, by the way, and TRV vector for this purpose. If you compare these viruses, PVX is a better virus for expression of bigger protein, where TRV don't have such ability for expression. It has limited expression. So this is our previous work where we have identified various genes which are involved in various functions. So gene function can be done. If you see that this is basically a gene which is responsible for the production of emanations in the cotton plant, and we have found via expression of PVX. So we use this vector. The plan was to express PVX in one vector, the CRISPR-Cas protein in one vector, and the guide RNA along with its scaffold in the other vector of PVX or TRV. So we clone the PVX. This is the methodology. We also designed the guide RNA here. The guide RNA which we designed were on the PVX gene, so that we can visually observe that whether that CRISPR system is working or not. This is basically the PCR amplification of the Cas9. This is amplification of the scaffold. Then they were cloned in the PVX. Here, this is the Cas9, and this is the cloning where we have shown the scaffold is cloned in the PVX. They were, infiltrated. We use two kinds of infiltration. First, we co-infiltrated PVX along with guide RNA, and in the next experiment, which is this, here we first infect the PVX harboring the Cas9. And then after 3 days, we infect with the scaffold of the guide RNA. So the result which we found was that in this case, we were unable to see any kind of bleaching in this case, which means that this particular thing is not working. But if you see these plants, we were able to see a little bit of photo bleaching of those plants, which means that Cas9 is basically expressing in these plants via PVX. Then in another experiment, we confirmed that by using the molecular analysis, by using RT-PCR. In another experiment, we developed a Cas9 expressing Nicotiana benthamiana plant, and we used our guide RNA, which were expressed from the PVX as well as from the TRV, and we have found the photo bleaching in this case, and it was really very successful. So these results basically tell us that PVX can express the Cas9 protein, but that expression is not up to the mark which is required for our experiments. So we need to work on the conditions for the expression of that genes. And here I have given you an example. This is a very old picture of my PhD study where I was working on the PVX. And what I did was that I was having an expression of one protein, which is basically the V2 protein. I put one tray in the glass house and the other tray in the growth chambers. The same gene, but the results were entirely different. That the temperature difference was 2 to 3 degrees Celsius, and the light conditions were different in the growth chamber as well as in the greenhouse. So from there we got an idea that these are the conditions, the light and temperature may play an important role in the expression of those genes. So we worked on that, and then we found that here we use a GFP gene in the PVX, and we use different temperatures and different light intensity. And we have found that if you change the light condition, if you go toward the lower light intensity, and if you go to the lower temperature, the expression of the GFP of the PVX can be increased. Here, if you from the GFP, you can see that. So it means that if we play with the conditions of the PVX, we should be able to get a better expression of Cas9 via PVX. So we are still working on that. In another the project, usually when you are working with these viral vectors, or if you want to express,
you have to finish, please.
These are 2 or 3 slides.
Okay, thank you.
For viral vectors, we usually infiltrate that. And this infiltration method cannot be used in the field. So what we did was that we have identified some conditions by which you can spray these agrobacterium. And for that, what we did, we used some surfactant, we used some adjuvant, and we were able to get, if you see this from here, these results, these are better. We were able to get the expression of these virus. The virus in this case was TRV. This is because PVX don't work on cotton, and these are the cotton plants. So when you spray on the cotton via agrobacterium, those viral vector can work. We are still working on these results at the moment because these are not up to the mark, because when we quantify the expression, it was very low, but we were able to see that. So we are now playing with the conditions to get the optimum expression. This is not the work of myself. There are several colleagues who are involved. There are several students who are involved. First of all, Dr. Shahid Mansoor, he's our group leader. Then Dr. Zaid Mukhtar, Dr. Shaheen Aftab, Mr. Muhammad Zuhaib Khan, Jawad, all are the students. The Jawad, Anila, Azka, Hamza, Kashaf, Ikra, Saptan, and Muteen. Thank you very much.
Thank you very much for your excellent presentation and also the speedy presentation. Now, our next presentation is on the iron biofortification through genetic modification in rice, wheat, and cassava by Dr. Seçkin Eroğlu from Department of Biological Sciences, Middle East Technical University, Turkey.
Hello. Hello, everyone. So I'm gonna give further details about one of the presentations today. So Emre Aksoy already told about this biofortification thing, and then I will give more insights about the three crops: rice, wheat, and cassava, the most important staples in the world, and how they were enriched with iron, and what problems were faced and how they were tackled. So you can see those three crops there. So cassava maybe is a bit foreign to you, but the other two I'm sure are quite familiar. So this map shows the micronutrient deficiencies around the world, and the take-home message from this, I think, is obvious. It's very widespread, and many people talked about UN aims, and one of the aims was to fight with hidden hunger. You can see the yellow color shows where iron deficiency is a risk factor, and green and black regions show not only iron deficiency, but where vitamin A and also iodine deficiencies might be risky. So what kind of, what happens if this risk is realized? What kind of damage will follow? In fact, for iron, for example, if the deficiency is low, then we have mild symptoms, like we may lose hair, and we may feel fatigue. I can see some people around back seats, they may have some iron deficiency. So if iron deficiency level is higher, then we have really more severe symptoms, especially in children. The IQ levels can remain lower. You cannot concentrate on your studies, and you cannot be successful. And especially this kind of nutrient deficiencies, such as iron deficiency, is very widespread in countries, in poorer countries where meat consumption is lower. And for developing countries, one aim is to fight with these micronutrient deficiencies so that you can use the full potential of your population. What kind of remedies are there in the market for iron deficiency? One approach, the primary approach, is to test for potential iron deficiency. For this, they check your ferritin protein levels in your blood. Ferritin shows how much iron store you have. If you have less ferritin, it's likely that you have less iron, and you may fall into iron deficiency very easily. And then after checking your iron, the doctor will prescribe you some tablets. These are just iron tablets, pure iron. It can be pure iron. And then if you use them, you will be fine. So why are we say this is a problem? Just take your tablets. But it's not that easy, because people don't use tablets. They don't like it. And secondly, organizing distribution of these tablets is very difficult. Maybe you have a government which thinks iron deficiency is a big problem, but 3 years later your government changes, and then all the efforts are gone. We need a continuous effort, which is quite difficult in developing countries. Therefore, some other people came up with a brilliant idea. They thought about, oh, we should put these tablets, we should not directly give them to the people, but maybe we should put them to something they eat. So this way we delivered it. And then what they came up with this idea, putting the tablets or iron into the flour. This is wheat flour and rice flour. I think we also have flour for cassava, but this is really a great solution, but still it has limitations, such as especially where rice is consumed in large part of Asia, people don't use that much rice flour. They just eat the rice. or in Africa, for example, one problem is they don't use cassava flour. They don't go to the market. In many rural places, the market is disconnected. So people just grow their own cassava, produce wheat flour from it. So they don't care. They don't buy their flour from the market. Therefore, these remedies have limitations. Furthermore, as an additive to flour, iron is expensive at the long run. Just imagine for the next 50 years, if all the bakeries, they put iron into their flour, it costs a lot. So as plant scientists, we offer them a much sustainable solution. Once you make the plant feed itself with more iron and load more iron to the seeds, then the problem should have been solved. But how to put more nutrients to the plants? The very first idea came to the mind: when the plant is growing, just put some nutrients in it, and then you will end up with more nutrients in the seeds. And it worked. It worked, but only for zinc. So in Turkish soil, zinc, it was extremely low in 1960s. We noticed this, and then we just put zinc into the fertilizers, and then we end up with much more zinc in the seeds. But it didn't work with iron. Doesn't matter how much iron you put in the plant, the seed will not end up with higher iron. A very big problem. So what's the, I want to tell you what the logic of this might be. So this is an analogy. When you have a fire, you carry water to the fire with a line of people, and doesn't matter how much these people work. If just one person is weak and he is not working well, then this guy will be the limiting guy for the water transport. This is similar to the plant. In the plant, iron must be taken from the soil, should be carried around the organs, and then should be put into the seed. A lot of guys are working, and these guys can resemble to the proteins. And if one protein is limiting, you don't have any chance. Doesn't matter how much more water you give from here, you will not end up with higher iron in the end. Therefore, what plant scientists must do in order to biofortify the plants is to find out who the lazy guy is. To know the lazy guy, we should learn who are doing what. And it's always complicated. I will give a brief description of how iron is taken up from the soil, but just believe me, a lot more is coming in the following years. Every year, this picture is getting more and more detailed. So this is the root, this is the rhizosphere, and iron must be taken from the rhizosphere into the plant by a protein called IRT. This is an iron transporter protein. But before iron can be taken in, because it's found as iron 3, and this protein cannot take iron 3, it must be first reduced by the plasma membrane protein FRO2. And there are some other helper proteins which solubilize this iron. Otherwise, iron is just staying there. So it first has to be solubilized, meaning mobilized, and then it should slowly come to the plasma membrane, reduced, and then only after that taken into the plant by this IRT1 transporter. And then once it's inside the plant, it must be always chelated in order to keep it solubilized. And we have the iron chelators, synthesizers, the genes that synthesize iron chelators, and so on. And don't forget, there are transcription factors controlling all these genes. Notable one is FIT transcription factor. FIT is not the only transcription factor. On the upstream of FIT, there are tens of other transcription factors. So FIT is here, and all this is here, but we have a lot of upstream transcription factors. We have, for example, identified the URI gene, This is a sensor. Wherever there is less iron, the gene is dephosphorylized, the protein is dephosphorylized. But if there's less iron, the protein is phosphorylized, and then it turns up all the downstream genes. So many scientists try to overexpress this gene, that gene, this gene, and then they will try to overexpress these genes, and then they try to find out the lazy guy. If they find, then they end up with higher nutrients. So all these information are basically found from two plants, rice and Arabidopsis thaliana. And then we just published this year a review about it, and we talked about the common approach to fortify the seeds with iron and what the logic behind is. I will just tell you maybe one of those. So scientists overexpress IRT1, and this didn't lead to any iron uptake. But then later on, we understood that the reason was the gene is under genetic control. Whenever the plant has more IRT1 overexpressed, it cleaves its own IRT1. So they came up with a solution. Let's put an IRT1 from completely different plant. Maybe then it will be subject to less control. And it worked like that. So they used, I think, apple IRT1, and then this then increased iron concentration inside the plant and even in the seeds. So due to time limitation, I'm not gonna further give details, but you can see there are a lot of strategies. Not any of them is perfect. That's why we have many. Let's see now some applications in rice, wheat, and cassava. So this is now coming from basic science to the more translational research. And here we can see rice on the top and also at the bottom. But so TG means transgenic plants. This is wild type, and all these articles refer to different approach. So the things to notice here is the iron level is higher in transgenic plants, came from 12 to 14, for example, or 4 to 13, like fourfold, okay? But it's not that much. It's not like 10 times. It's not like 20, 100 times. And this is very low, in fact. And another thing you can notice is there are so many studies. So a lot of people are working on it and have limited success so far. Okay, another thing you may notice is whenever you have more than one strategy, you end up usually much more iron. For example, here 14, it became 4. It became from 4 to 18, like almost 5 times, when more than one strategy is used. Why is it like that? I told you there is one lazy guy, but in fact there are more lazy guys. So whenever you spot one and replace it, you will end up with again some other limiting guys, and you then have to change those. Okay, that's the logic. Another thing you may notice is how nicotinamide gene is used. Nicotinamide is a chelator I mentioned to you about. Whenever you have more nicotinamide, it worked with many plants very well. More iron goes to the seed somehow. Okay, this was about wheat. And now we have, sorry, that was about rice. And we have now wheat. Here with wheat, we have much less references, so much less attempts. We again have different genes, and you can see more than one gene, I mean, and you can see again, sometimes rice genes are used and genes from other plants are used. Why? Because we don't want plant to to use its own systems to degrade the protein. Okay, so I'm telling this specific example, but many things I tell you, I mean, you can find parallels with other genetic approach. Okay, so this was wheat. Sorry, this was wheat. Okay, where is cassava? Okay, I think, yes, cassava is here. So cassava is here. With cassava, we had very bad days. So you can see there were some studies, but they were never published because the increase in iron concentration was very low until Narayanan, our teammate, make a big breakthrough. So I don't know if you can see, but he just expressed Arabidopsis, IRT1 and ferritin. So IRT1, remember, is the protein takes up iron in the rhizosphere, and fer1 is like human ferritin, it just captures iron. When these two were expressed together, let me tell you, the iron concentration went like 20 times, which was really great. Okay, so with cassava, although we had very low background, we achieved really high levels. I hope we can also reach those levels with rice and wheat in the future. And now another critical thing. Some of you can ask, okay, we already have solutions to micronutrient deficiency problems, like we can put iron to the floor. And this kind of approach, we pay so much money, and these plants will be GMO and people will not eat it. So why do we do that? Okay, now I want to take your attention also to here. Here is again, IRT1 and ferritin is overexpressed, but there's another gene, CBSD, which is nothing to do with iron. It is against a viral disease. And this is used for cassava. Okay, the logic of biofortification approach is not to just biofortify food. I think we should look from a bigger perspective. We want to create super plants that has much more nutrients. Not only that, but more resistant to diseases, more resistant to drought, and so on. So we are not gonna put them to the market just nutrient-rich plants, but in the near future, plants that have superior properties that people want to adapt. So these are African people, and this is the cassava Narayanan produced by overexpressing IRT and ferritin. It looks like just a regular plant, but it makes great changes in African people's lives. So at least some of those poor people will have higher IQ and probably end up in good universities and become a very, will be a brilliant, will have a brilliant future. And poor people in Pakistan, in Turkey, they also like greatly from deficiencies. Maybe someday we achieve really high iron levels in wheat, which may lead to a big change in their lives too. With this, I want to finish my talk. So we need better understanding of bottlenecks in nutrient loading from soil to the seeds in order to be more successful in the future for biofortification. Secondly, we found most of the things in basic crops, but for every crop, it's a bit different. So we need to transfer the knowledge from basic sciences to the more stable crops. And thirdly, very important, another aspect is we need to have better adaptability for GM crops, because even if you have very nutritious crops like golden rice, for example, due to the opposition, it may not end up in the market. And we have to tackle this issue as much as we have to tackle the scientific issues. Thank you very much for your listening. I think this was the last talk, so maybe we can even get questions. I don't know.
Yeah, thank you very much for your nice presentation. Now we have a panel discussion and question and answer session. So if we have questions, you can ask question to any participant in this session. And also we have offer, I think we are also online, is it? Imran? We have also online participants from different countries.
Yeah.
Yes,
sure,
okay,
fine.
okay.
No.
I think we don't have any question right now, so we'll postpone this session for the next session. And it will start at what time? 30, it's 15,
3:10.
yes, 3:10. So in our next session, we have only 2 presentations. So we will definitely start from this.
Yeah, I would like to, would like to thank all the speakers who did a marvelous job for delivering the lecture from basic to the translation, everything. Okay, thank you.
Thank you very much.
I would request all young people like me should be here in the hall. Tahir, जो लोग बाहर खड़े हैं बेटा इनको कहो अंदर आ जाओ. So that and Dr. Saab work on some difficult crop for tissue culture. So I hope that it will be very enlightening for all of us. Yes.
Thank you very much. I feel immense pleasure to be here, and I really thanks the comstech and all other organizations which invited me to present my work here. At first, I changed. I had a different topic. Then later on, I changed this topic to the innovation in plant tissue culture. And before I start my work, I present what is innovation. When we say innovation, innovation means to something new, creative idea. Or you have already some work and you want to improve it. So when these both combine together, there is a novelty. So in my presentation, I will start from the plant tissue culture. Okay. Okay. I will present the general overview of plant tissue culture, not too much in detail, just I will go through from my slides. Then I will discuss the problems or challenges in plant tissue culture, which is very common in both in the lab conditions, if you are working to optimize the protocol for commercial propagation or for the for some modification like genetic modification, you need a proper and repeatable protocol. And in this last stage of my presentation, I will give some slide. I will show some slide about my recent works, which is related to some artificial intelligence. So I used this technique in my plant tissue culture work. So what is plant tissue culture? It's just taking an explant culture on the synthetic media, placed in the growth room, and you can get the plants. So what will be the procedure? Is it you want direct or indirect regeneration, mean organogenesis or embryogenesis? It depends on you. So what is the difference between this direct or indirect regeneration is basically the callus. If you are going through the callus culture, it's indirect regeneration. If you don't want callus, then you will go through the direct organogenesis or embryogenesis. Another important point is what type of plants you want. You want auxiliary or adventitious shoots, and it depends mainly on the explant. The stages of plant tissue culture. It's a very debatable point because in the literature you can find 3-stage plant tissue culture, 4-stage or 5-stage, but all the stages are same. And that is the explant starting from the collection of plant material to the greenhouse condition or the field conditions. The first one is the culture establishment. Second is the elongation and multiplication, then rooting, and then the acclimatization. Sorry. The first stage is sterilization. The sterilization is one of the most difficult stage in my point of view, especially if you are working a material which is directly taken from the plant, like twig or the bulbs. You have to follow some difficult procedures to clean the material. So it depends on the single or multiple step procedure. Here one is the picture which we recently developed a sterilization protocol in pistachio rootstock. In the second stage is the in vitro culture, which started from the post-sterilization to the subculture and up to the rooting. So this is the second stage, and the third stage is the rooting. Do we need rooting in vitro or not? That is the debate because I will discuss later on. In most of the studies, now we can skip this point. There is no need of in vitro rooting. I will give you some examples. Do you need some hardening or some other points or some other things in this stage? Here there's some picture which I done in my research work is a chickpea, and this one is the pistachio root culture, sorry, in vitro rooting. In the last stage is the acclimatization. We need some steps to acclimatize plants. We need growth rooms, growth chambers, or greenhouses. This plant is the chickpea, which we used acclimatization in the growth chamber. And then, or other, we need a simple growth greenhouse. And this is picture from the mulberry, the commercial propagation of mulberry, a recently finished thesis by my student. What is the application of plant tissue culture? Very common. You can divide this into commercial propagation or propagation for the breeding purposes. So I'm not going through this because it will take time, but there are some challenges. Either you are doing work for the commercial or for the breeding purposes, you are facing some problems. What type of problems? These are the problems in propagation, like the browning of plants in the tissue culture, contamination, this endogenic bacterial fungal contamination, and then plantlets, burning of the plantlets, delay of subculture, very common in the commercial propagation, some type of somoclonal variations, and then in the rooting difficulties. Other is the abnormalities or some other disorders which can find out, or you can look in the tissue culture. And one is the hyperhydricity, a very common problem in the plant tissue culture plants. And then is the recalcitrance nature, very common, and one of the biggest challenge. If we cannot optimize the protocol, we say is this plant is a recalcitrant. And then albino plants, shoot tip necrosis, in vitro habituation, the plants grow in the medium if you are not providing some type of hormones, swollen base shoot, callus culture, and yelling or the leaf excision are some common problems in the plant tissue culture. Here is an article recently published which address all these challenges and their remedies. So if you follow this article, you can learn a lot about these problems. Now, what is recalcitrance? Here are the stages. Which stage is the more recalcitrant for regeneration? In my opinion, each and every stage is difficult. Some plants have sterilization problem, some plants have regeneration problems, some plants create problem during rooting or acclimatization. Even some plants we can face 2 or 3 recalcitrant problems at the same time. Explants. Then sterilization problem You cannot find, for example, when we used sterilization of pistachio, we started sterilization, we finished, there was no contamination, but even there was no regeneration at that time. Later on, we found what are the reasons, and when we overcome the issue, we got a big success. Then organogenesis or morphogenesis stage is also a challenge. Rooting, a big challenge in legumes, especially chickpea and lentil. Then acclimatization problem. And we can overcome this issue by changing nutrient mediums, culture conditions, or just change the explant. So we sorry for the disturbance. Here you can find out a picture in which it's looking a good picture, but these are not the normal shoots, they are the abnormal shoots. So what type of explant we need to select? It depends on you, either you are going for the axillary or adventitious shoots regeneration. In my opinion, we can use the seed or zygotic embryo as a seed explant, or you can use a combination of preconditioning or post-conditioning combination. It means you can, if you have a sub-explant which is recalcitrant, you can give a shock to the explant by giving it a culture on the high cytokinin medium. Like you can use BAP, benzene aminopropene, at 5 to 25, even up to 50 milligram per liter, but depends on the duration, how much long you can use it. But a shock activate the meristematic region, cell division, and when you transfer these plants to the new generation medium, which may be devoid of any PGR or low BAP concentration, you can get an excellent result. And I use this protocol in most of the legumes, in chickpea, in lentil. I use this protocol in cowpea, in groundnut, and I got 100% regeneration by using this protocol. And even there's no problem of rooting. For example, another advantage is if there is a explant is very small and you cannot isolate it, just grow on the high cytokinin medium. Within few days, it can elongate and you can extract or isolate your explant easily. Here, another example of I use the seed directly on the culture medium, and we use 3 different types of PGRs. A and B belong to BAP. This one is the kinetin, and the last one is the thidizoran. And you can find the difference. Each explant almost regenerated multiple shoots, but these all shoots are not same. So there is a possibility you cannot get the desired results from the same explant. So combination of PGRs is also important, and their concentration is also important. In this, it was abnormal multiple proliferation. When we transfer this plant to the medium devoid of T days, it proliferated a lot, but these shoots were not ideal for the rooting stage. I will discuss it later on. I used this seed explant in industrial hemp, and I got multiple shoots from that explant. And then later on, I just checked, can I use it for the genetic transformation, and that is the result. So on the basis of this, I just proposed a project on industrial hemp for genetic transformation optimization in Turkey. And this one is the seed explant, and I got multiple shoots of sorghum directly from the seed. So rooting, in vitro rooting, is, in my opinion, is one of the difficult stage in plant tissue culture. And at that stage, the selection of proper shoot is very important. The shoot which is very near to the natural conditions in the nature, it must be hardened. If you select some soft tissues or hyperhydric plants, it will not root. The root will be a problem. But in legumes, we are facing some problem. If we convert, we culture these plants on the rooting medium, there's also chances of multiple shoots. Expect even there is a rooting here, but it's a callus development, and then it's the multiple shoot. We are also facing this problem. So in this experiment, it's a chickpea. I cultured these plantlets on the high sucrose rich medium, and I got 100% rooting in that plant. And this is another aspect in which you can directly rooted plants, the individual regenerated plants in the soil. It's a special designed Van der Nepp soil for the plant tissue culture for the commercial propagation. You just skip, and you can imagine how better it is for the commercial propagation that you are just skipping the whole 1 month process. Just take the plants and put it in that in the soil, and you can got the double impact of rooting and adaptation at the same time. Now we are working on this to use this type of soil for our legumes and other crops. Can we use this for that plant? If we succeed, I think it will be a great success for in the future. Then acclimatization. For acclimatization, this is a picture of the walnut, the commercial propagation of walnut. The regeneration was very easy, not easy, but easier as compared to the other plants, but its adaptation was a big challenge. So our team developed a new protocol in which we use the multiple greenhouse condition at the same time. For example, here a worker is cleaning the shoots directly taken from the in vitro, then put in the soil and place them in the trays, and then cover it with this polyethylene, and these bags are placed in the greenhouse. So this is the outer look of that greenhouse. Inside these are the trays with plants. And if you check at above, it's the actual greenhouse. So it's a triple greenhouse effect, we can say, the triple layer, three layers. So in this way, what was the end result? You can see here, we got the high level of acclimatized plants. So this is also a new way of rooting plantlets under especially the commercial propagation. Hyperhydricity or abnormal shoots is one of the greatest challenge. I already showed you this picture. It depends on you can change the culture conditions, especially the hormones. You can change the agar, you can change the screws concentration, and in this way, you can overcome. Because if we get this type of plants or these type of plants, we cannot root easily. These type of plants generally lead to the callus proliferation, callus induction, and this is not this callus is not a desired characteristic during the rooting. Now I come to the last part of my not last, actually the most prolonged part of my presentation. So what are the latest advancements or the new technologies adopted in plant tissue culture? There is the bioactive compounds, a very famous now, most of all over the world, you can get a desired amount of our phenolic compounds or the metabolites from this using plant tissue culture. And then you can use the nanoparticles in plant tissue culture for different objectives. Especially this plant tissue culture is now using for the food products and the cosmetics industry. The use of bioreactors is very common. Then endophytes in plant tissue culture. There are change in the growth rooms or chamber. There are special design chambers for the special plants. I give you some examples. Then these two plants, these are the statistical analysis, the model analysis using machine learning or artificial intelligence based. I will give you some example on that. And then the automation, the robots automation is now involved in the plant tissue culture. So when you say this metabolites or secondary metal, the bioactive compound, is the organ culture you can use. You can add precursors in the plant tissue culture medium. You can use different type of elicitor, physical or chemical, biotic or abiotic, biotic elicitor like fungi, bacteria. You can use hairy root culture, endophytes, different type of bacteria and fungus are available which can use for the elicitation of the compounds. Then the commercial bioreactors for hairy root culture. Secondary metal immobilization and accumulation is also done by plant tissue culture. Metabolic engineering you can also involve during, and then some in vitro mutagenesis. It's also very common nowadays using physical or some chemical mutagenesis directly treated with these mutagens. You can culture these plants in the plant tissue culture. And this is biotransformation, and The most common is the genetic transformation. These are the areas where we can work by plant tissue culture. It is an outweigh of different plant tissue culture techniques. Here are the two research articles, the review article recently published in Engineering in Life Science, and in which they develop a protocol for food products. For example, in this slide, you can see that is the citrus vesicles under the electron microscope. They use this bioreactor, the scheme to produce. Now, this is a way you can, maybe in the future, you can find some food ingredients through plant tissue culture. Now, these are the modern growth room chambers. Here, we are not using the fluorescent lights. Now, we are using the LED lights. The combination of blue and red is the most commonly used combination is 3:1, 3 reds and 1 blue combination. It gives a pinkish appearance. Or we have some specially designed chambers like this chamber was developed for the hemp production. And this picture belongs to the microtuber production of potato using the plastic bags or some type of bioreactors. So within a small space, you can produce a large number of microtubers using this technique. This picture belongs to my old laboratory in which I developed this different LED lights for checking the impact of LED lights on in vitro germination or even ex vivo germination. Also, we checked did some experiments on that regeneration and got the excellent results. The work is still not published, unfortunately. Another important area is the nanoparticles. So in nanoparticles, its application is in almost all phases. This is the sterilization. For example, the silver nanoparticles are used for sterilization, also used for enhancing germination, organogenesis. Nanoparticles are used in the genetic transformation. Some types of somoclonal variation can be achieved by nanoparticles. And then secondary metabolites are very common nowadays using this. And also we can enhance the rooting frequency by using nanoparticles. There are some articles recently published on the nanoparticles by my group. But don't forget, there's also a chance of nanotoxicity in another area. So we must focus on the exact concentration for the desired targets. Another important area is the endophytes. These endophytes are the bacteria and fungus which produce auxins and gibberellic acid, and you can use these endophytes in the tissue culture. So there is no need to use the external provision of these hormones. So in this way, you can easily multiply the shoots. Even these endophytes also help to for the rooting and in the acclimatization. Here you can see easily. Now I come to the last part of, and that is the application of machine learning or artificial neural network or artificial intelligence in plant tissue culture. In this slide, you can easily say, when we perform an experiment, we go for the classical statistical analysis or ANOVA. And another way is that you can get or validation of your data or optimization of your data through machine learning, or you can use both analysis at the same time. So what is the design of experiment? It's also, there's a technology called response surface methodology. It's also the earlier part of this artificial intelligence, in which, is it clear? Yes, okay. In which you can analyze your data and you can place your data in the 3D dimension. So it's called the surface plots. So it distributes your data on the surface and you can check your data from any angle on the computer program, or you can use contour plots to distribute your data in subclasses. In which area you want to go, you can select the combination here. But to make this data, there is a need of minimum 2 input variables. In this way, here at the last stage, we ask the computer, that is my target. And I ask, for example, I want a maximum regeneration. What is maximum regeneration? 100%. But I give an option of maximum germination. The computer asks me, okay, you can ask me 100%, but I can give you a maximum of 98%. And these are the two combinations which can you use, go perform the experiment and optimize it. So this is a new methodology which we are using. But nowadays, another important aspect is that now you can use this procedure prior to the experiment. You can design your experiment with the help of computer. You just give the input variables, the amount, whatever you want. It will design an experiment for you. And when you give the results, output variables, it will give you the different options. What do you want? You can directly optimize your target. And for this, when I just said I was working on another topic, at that time I had a plan to give the full information about this. So now I'm just going through this slide. So another important area is the, I have time, I have to move.
Please continue.
Okay. So that is the application of artificial intelligence in plant tissue culture. You can apply it in all fields of agriculture, sciences, or biotechnology. An article published in 2020 on the artificial intelligence in all fields revealed that only 3% articles belong to the agriculture sciences, and it was the least. What is the reason? Because it's a pure data science. You need some data scientist who can manipulate your data, who can work on your data. We are plant scientists, so our computer knowledge is up to a level. We cannot go beyond. So in this aspect, I consult with my friends. Two of my friends from Turkey working. One is working in the Bilkent University, the top university of Turkey. So we were now working together. Here is an overall. When we use these terms, actually, the basic is the artificial intelligence, and within the subclass is the machine learning. Machine learning is the algorithms. You can use different algorithms, and then in machine learning there is the artificial neural network, which is commonly known as artificial neural network or the deep learning. The deep learning is actually the image analysis. It's a classification. You can use images to analyze your data. Another aspect is there are three different types of machine learning. That is the supervised learning, unsupervised learning, and reinforcement learning. This supervised machine learning is very common in our field, in plant tissue culture. We can, or in plant biotechnology, we can use this supervised machine learning. Here we can say we can use, and in supervised machine learning, the two most commonly used methods are the regression or classification-based models. Here is the overview of the whole machine learning process. It's a data collection, then data preparation. You have to select a proper model or algorithm. Then you have to train your model, evaluate the results. Then there's a need of tuning of the parameters, and then at the end, it will make some predictions. But before that, we have to scale our data. We have to convert, we have to work with our data. Because, for example, if I say, I got 4.5 shoots, I have to convert all the input variables into a spatial scale. And after that, we use, so for scaling, we have different techniques, we use different techniques. And then there is the use of hyperparameter optimization. There are three different types of hyperparameter: grid and randomized search and AutoML. These AutoML are the latest and very famous nowadays, this algorithm. These are used with the models, and recently we used this genetic algorithm for the optimization of tissue culture of industrial hemp, and this Firefly algorithm for the phytoremediation studies, and we got excellent results. And at the end, it gives you a combination of input variables. You can use these variables and validate your data. Then there is a need of cross-validation. Either your data is wrong or right. And the most commonly used, the outputs are the R-square, the R regression coefficient. But there's the most commonly used the regression coefficient. And then we use some other, we call this the performance metrics. This is a mean absolute error, mean square error, and mean absolute percentage error. There are a lot of other performance metrics. You can use them. So it is better to use more than one performance metric. For example, if you have a low R-square, the R-square is generally lie between the 0 to 1. 1 means 100% your data is accurate. But if, for example, if you get 0.1, it means, it doesn't mean your work was not good. Your work was not good. It means you have to check other parameters. Then there's a classification methods in which you can classify your data, and then you can use, in which the most important is that it distributes the data into true positive, true negative, false positive, and false negative. And then it makes 4 different types of methodology you use, and you can check you can validate your data. Here in this slide, okay, I'm finishing. This is slide taken from the application of machine learning in plant stresses. So here you can classify your data, you can quantify, you can identify and you can make some prediction using this. Here are some working which we recently published on the machine learning, in which we used 6 different models. The 5 models belong to the machine learning, and this one was MLP, belong to the artificial neural network model. And we got an excellent results, and this paper was published in 2022. Another paper recently published in genetics, frontiers in genetics, in which we used also, here the regression values were very low, but we use other parameters, and these parameters exhibited the better performance of all the models. Here is the presentation of whole the data on the one line, and this is a confusion matrix we made. This was the recent application on the published industrial crops and products on the in vitro germination optimization in industrial hemp. And this, which we use a different approach of classification model. We got our data and we set the threshold level, and then after we got these results, and these results suggested that our model predicted over 100%, almost 90 to 100% the data is accurate. These are the work which I already submitted for publication. This was on the phytoremediation studies, which can also use. Here, the most important thing is, again, we use this RSM technology to optimize the best performance. And we got that if you want to 100% cadmium removal from the water, you have to use 3 milligram per liter for an exposure time of 120 will give you the exact 100% removal. And then work on the chickpea, we are already submitted. And this is the new way of transportation, automation of plant tissue culture. And this one is the last slide, the use of robots. So there is no role of humans in the future. and one thing, if I can play this, let me check. I think it's not. So this was an excellent video in which I cannot play here. The uptake of explant from the medium, cut through the laser, and then culture again on the medium through the robots. So this is a new technology involved in the plant tissue culture. Thank you for listening to me. And I do extra time.
Thank you very much, Dr. Sab, for excellent presentation. I think that we will have a question at the end. So now we can invite Dr. Shaheen Aftab. She's working at National Institute for Biotechnology and Genetic Engineering. Madam, please.
So I would like to thank first the comstech for inviting me and providing the chance to share some of the work done at NIPCHE. So starting from my talk will be focused on the development of abiotic stress tolerance sugarcane by genetic engineering. So keeping just a very quick, overview of the sugarcane, Pakistan is the second major cash crop after cotton. And the major constraint for the production of these are the biotic and abiotic stresses, including the drought, salinity. Thank you for the site. For the crop improvements, for the biotic and abiotic stresses, you need a gene source, either plants or synthetic. So in case of the sugarcane require a 16 to 20 irrigation, and if water stress applies, so that photosynthetic activity is reduced, and you can get a stress plant and cannot get the yield properly. Another factor of abiotic stress is the salinity. If you increase the salinity, the production of the sugarcane is reduced if the EC is 18.6. So our aim was to transform sugarcane varieties, and we use 4 varieties by transforming this stress tolerant gene, ATNHX, AVP1, REV1, and then evaluation of the sugarcane under pot and field experiments. The prerequisite for any of the genes, you have to be transformed either using the genome editing. You have to go through and establish the well-established regeneration system, and we established 4 cultivar regeneration system at NIPG. These are the functions of these genes which we used for transforming the sugarcane varieties, and this is the transformation system taking from the X plant, getting the plant, and then internal and in field. So this is slide shows the transformation efficiency, and it varies in different cultivars. After development of the sugar or any of the crop through transgenic plants, first you have to analyze using the molecular analysis, and most of the techniques for the molecular analysis we use PCR for presence, transgene, Sadram for the integration of the genes, and then for the expression for northern or qPCR, whatever you want to use. And similar in case of the ATNHX, these are the same, and you have to perform with the third one gene is the REV1. So route study, before putting them into the field, you have to analyze these plants at the lab level or basically in the pot studies, and we used 3 levels, 100, 80, and 60% of the water supply in route. And phenotypical difference between these plants, you can visually see between the 100% and the water stress plant, and this is the root in the transgenic plant and versus towards the control plant. So effect of the drought on the cane height or plant height, you can see these are as comparable to the control. These plants perform better under the drought stress condition. So this is the photosynthetic rate, and it is very obvious in the transgenic lines as comparable to the control line. And in case of the ATNHX, we expose these plants for the different level of sodium chloride and measure potassium and sodium content in these plants. This is the pictorial view in the pot experiment under the tunnel condition, and when we grew them these plants in the different salinity level, and this is the pictorial view difference between the different level in the growth of the plant. And then we measure the sodium content in the transgenics as well as in the non-transgenic plants, and you can say there is a significant difference between the transgenic and the non-transgenic lines, accumulation of the sodium, and similar in case of the potassium. And then screening of these REV1 genes for the cold tolerance was carried out at 4 degrees centigrade after multiplying under the in vitro condition. And 3, 5, 7, and 10 days of this was given, stress was given at 4 degrees, and then cell membrane stability was determined. And this is the pictorial view. You can see the height of the plants versus transgenic and non-transgenic under the cold stress. And then we measure the electrolytes release in case of the control and the transgenic plants. It is obvious in the all non-transgenic lines releasing more electrolytes as comparable to ton. And then we perform the field evaluation of these plants. This is the view of the REV1 genes under field condition at RE Faisalabad, and this is the recovery of the sugar, and this is the yield, and you can see 4 to 12%. I mean, is a 4.8% visual difference between different lines, and 12% is the maximum. And again in the recovery. This is the pictorial view of the field experiment with the REV1, no sorry, the AVP1. And then you can see the difference between the 3 of these irrigation level, and increase in the sugar recovery as well as in the yield comparable to the non-transgenic controls. So this is the 18HX plant under the salt condition at Pindibhatiya. So this is the data. And again, I mean, the transgenic lines are behaving better as comparable to the control. The data, this is very recent data obtained from NIA under the natural condition. And they performed this experiment at their field. And 3 of these lines, they are performing, and their performance is outperforming at these locations. And these are the agronomic character. And this is the qualitative traits. And obviously, I mean, you can see is the bricks content, and then is the sugarcane, and this is the yield. So before commercialization, you have to pass through the biosafety studies or risk assessment studies, and you have to be performed at 3rd level party. And we have a lab at NIPG toxicology lab. They have performed these studies, and I didn't put a lot of the data, but I just summarize only 1 or 2 slides. And this slide shows to the blood biochemical analysis of male rats after 90 days of treatments of AVP1 transgenic sugarcane juice, as well as leaves. And there is no difference between any of the biochemical parameters between the transgenics and the non-transgenics. And similar case, you have to do the nutritional composition of these transgenics and non-transgenic composition and find the unintended effects, any of them. But we cannot find any of these unintended effects of these, and all of these approximates, they are almost the same as the control one. So I would like to conclude my talk with these words, that transgenic sugarcane lines, which we develop are drought, salt, and frost tolerant under the stress as well as normal or natural condition. So the subchronic studies shows non-toxic effect of AVP1 and other transgenic sugarcane on rats. No significant difference in the nutritional composition of the AVP1 transgenic as well as other 2. So I would like to thank Director NIPG, Head ABD, Dr. Zahid Maktoum, Dr. Shahid Mansoor, all lab members, researcher of the projects, MPhil, PhD students, collaborator, SIR, and NIA, toxicology lab members, Pakistan Agriculture Research Board, and HEC for funding. So I would like to say, if we only 20% irrigation we block. So the economic impact of these with the yield 2% increase at 10% adoption, the economic impact of these transgenic should be more than 2 billion. So thank you so much.
Thank you, madam. I think that we had a wonderful session. So please have a seat. So Imran, shall we invite all the speakers? And, we can start. Asim Saab, I think I needed you yesterday. It was so good. So please join. So ladies and gentlemen, and all the colleagues, so this is now the point for discussion. I think that there must be a lot of questions and comments. So this is the time that we have this discussion. So
Yes, Imran, maybe first question. Please wait, the microphone is coming.
I want to ask from Dr. Asim. First of all, it was very nice presentation. I mean, you know that right now this speed breeding is a reality. What about speed transformation or speed in tissue culture?
Okay.
Can we apply the same technology on that, and would it help? What are your thoughts? I'm not sure about
yes,
just an idea.
The time is very important factor right now. So in tissue culture, if you go through the callousing, it will take a lot of time. So you have to go for the shortcuts. And that is my hobby, to apply the shortcuts in plant tissue culture. I just present one picture on the genetic transformation of this hemp, industrial hemp. What was the idea between that? If you check out the embryo, if you use embryo, what is the, which part of is the meristem is the embryonic axis. When this seed grow, it convert into cotyledon node, and cotyledon node is also meristematic region. So no need to grow the plant. Just when grow this plant, this seed on the tissue culture medium, when that part is exposed on the culture medium, at that time you can inoculate with agrobacterium and other method. And we did the same in that plant, and that was just one example. The efficiency rate was very high, I can say. In hemp, and hemp itself is a recalcitrant. Why? The rooting is a big challenge. Regeneration itself is a big challenge for this hemp. Okay, so in this way, you just hit the exact point of embryonic axis that it's exposed. At that stage, you know, the meristem is active, so it can update. It will be good for you, and there is no need to go for the multiple shoots, even the single shoot, and then transfer them to the soil, and you can get the plant, in my opinion. Another point is the speed. If you want to go for this short time regeneration protocol, a full development protocol, I use this preconditioning and post-conditioning combination. I give 1 week to 10 days, a combination of 5 to 20 milligram per liter high concentration of cytokinin. And in this stage, you can get 2 to 3 shoots, even in some explants. Just transfer them to the MS medium and you can get the shoots. So no need to multiply further.
Okay, thank you. Let's wait a little bit for maybe students to raise hands, and you can also join from the internet. Yes, Oppo and others. No question from the audience. Maybe they can write, if they write. Yeah, maybe if you write in chat, then we can receive your questions. Okay, the internet people. Okay, from the audience, I see one hand. Yes, please.
You were asking?
Sorry, there's one person there next to you. Okay.
So a few days ago, a purple tomato engineered with high antioxidants was approved by USDA. It has capability to fight cancer and prevent cardiovascular diseases. And USA has approved its cultivation in spring 2023. I want to ask that what could be the possible decision of OIC countries on cultivation and commercialization of purple tomatoes? If we don't, then why? As we know, Pakistan has high rate of cardiovascular diseases and cancer. If we have a preventive measure now, then why will we refuse it? If I also want to know the thoughts of distinguished panelists on purple tomatoes. Thank you.
This question for you, Sachin.
Okay. I think that, you know, this is a very good question, and I fully endorse that, you know, such type of products should be commercialized in Pakistan, and maybe so the decision to commercialize or not, it basically lies with the government. So I think that, you know, as a representative of Pakistan and NIFG, I think this is some of the things that we should, you know, so biofortification, as we discussed in the morning, is a very important area that, you know, you instead of giving medicine, you know, if you have enriched plant with for certain nutrients. So if you go and search, there are other, you know, nutrient like vitamin D by a group in Donini Center. And also, so there's so much going on. So I think that, you know, these are the things that we need in the country. There are normally some issues in introducing, either that, you know, they ask for the what they call as technology fee. So, or maybe in friendly countries, you know, you can get it some of the material free. So I think that, you know, that's something that we look forward. We have already contacted for some of those biofortified products, And we are very keen on this. So yes, good idea.
Okay. Thank you. Normally it's your turn, but the microphone is there. Okay. So, yes.
Assalamualaikum. I have a question, general question. I have visited Niqab, Islamabad, where I have seen many researches going on the genetic improvement of the crops. I have also attended the conferences of where scientists from NIPG, Faisalabad, were presented their successful researches. Like today, Dr. Shahid and Dr. Imran share practical examples, but these are just work for publishing. As Dr. Laila said that the Pakistan has only one GMO crop, why are we lacking in commercialization of these GM crops?
Well, I think that, this is a matter of, a constant struggle. There were some efforts to commercialize GM maize in this country, but, at the highest level, it was not allowed. So that's why, as an alternative strategy, we are moving towards a genomics selection or genome editing because, there's no foreign gene. So it's something that, we all should struggle, but obviously the decision lies. But there are some guidelines in this, for example, so I mean, obviously, we started to work with rice and wheat, other food crops, but there's a difficulty commercializing those crops. But I think one of the things that we can do is that, focus on some of the crops which have been commercialized in other parts of the world. I mean, I know that, maize is already commercialized, but like, oilseed crops, sugar crops, and cotton. So those are the crops that we can probably at the moment we can focus if we are working on genetic engineering. But for genome editing, what we are hoping is that there should be less of an issue. These are the things that I can say that. But we have gone through all those processes of developing products. I remember that in early days we developed NIPG developed transgenic basmati rice and CAMP developed transgenic rice for BT or bacterial blight resistance. But those were not accepted by we were not allowed to commercialize those. So these are country policy, but it's not wasted. It's actually you learn how to do all those transformation work.
I would like to add is that we have conducted this field trial of getting the National Biosafety Committee approval on sugarcane for providing them the data on biosafety for the commercialization.
Okay, next question. Thank you for your patience. Yeah.
No problem. I would like to ask Dr. Asim.
Yeah.
First of all, nice research, because whatever the transformation or GMO development, it's all is based on tissue culture or regeneration. I would like to ask about the color light.
Yes.
What did you found if we use that pink light in comparison to the white light? So there is a difference in the wavelength or intensity, and then how can you determine that particular wavelength, mean pink wavelength or red or blue, or it's just to optimize?
Okay. Is it okay? Your question is over or something you want to
And
add?
then I would like to ask that how much difference did you observe that in comparison with the white light, you are getting 5 plants per explant, and here you're getting 10, like 15 or.
Okay. First of all, these LED lights, their age is very long. You can use up to 50,000 hours. So that is one advantage of using these LEDs. Second, if you go to the photosynthesis, we need red and blue lights. Mostly at that time, we used a range of photosynthesis. So in this way, you are just providing these lights. So, but it depends on the plant. The research conducted in the greenhouse and in the in vitro, every plant has a different requirement. You cannot say, but in the commercial propagation or in the greenhouse combinations, this pink light is due to the 75% red and 25% blue lights. And now it's commonly used in the greenhouse conditions. This is now confirmed for this tomato, for cucumber, and such type of greenhouse crops. But in vitro, we cannot say that this combination is the best. You cannot say. I'm giving you one example. We used this black chickpea germination experiment. It's very speedy plant, normally germinate in 30 hours under in vitro conditions. We used 5 different light combinations, and under green light, we got 100% germination in 18 hours. That is the difference. Black chickpea. So, yes, please.
So can we hypothesize that if X plant is green, then that pink light would be more effective, and if X plant is non-green portion, that pink light.
It depends on. It depends. You know, in tissue culture, in my opinion, there is no protocol. Today, protocol is working. Tomorrow, it will not work. You know. Yes, please.
Sorry, before that question, we have a follow-up question from internet. They ask, in technical terms, how much heritability was observed for tissue culture and utilizing machine learning for it? Maybe reproducibility, or I don't understand.
Actually, we didn't work on this heritability right now, but we have one work on this machine learning, in which we use data from another group, and we just analyze the results. And but you can use heredity experiment by machine learning. It's now very common right now. Yes, okay. It's possible.
Yes, thank you very much. Yes,
okay.
Allah Baksha.
My question is from Dr. Shaheen. Thank you so much. Very good presentation. Our success is really dependent whenever we take product to field. So many congratulations. I have quick queries. These constructs are independent transgenic?
Yeah.
Okay. Second, what about have you established their zygosity level, keeping in view the polyploidy level of sugarcane?
Actually, in Pakistan, there is no flowering. Okay, so it is totally micropropagated plants. No flowering, not going to be a transfer to next generation. You have to plant them and then harvest from the same plant. There is no seed formation, flowering and seed formation, no crossing, no zygosity. Whatever you add them, they will remain there.
All right, I got it. And partly you have also already replied. So what is its current status right now? Have you applied for its commercialization license?
No, not yet, because we are.
And also, but you told you have got the permission for field trials, right?
Yep.
Okay, and please.
The recent is that our one line is under new trial, and these 4 lines which I have mentioned just here is the data comes from NIA 2021. We're still getting the more results, and Tahir is here, and inshallah we will put these lines also in the new trial, and then we'll be able to get the data, and then for commercialization. 2 years data we need.
Okay. Oh,
thanks.
Trial. All the best.
Thank you so much.
Okay, can microphone go to the top? Okay, next here. You are also.
Yes.
Okay.
My question for all the panelists. Is there any global recognized ethical code and morals for how far or how much to go further on genetic reproduction or regeneration? Because I have seen that pesticides and drug producers are utilizing this tool to favor their commercial benefit, limiting the harmony of the seeds, not for the favor of the human being, but favor of their commercial benefit. Who will set the rules of code of ethics and morality? And who will accept this code of ethics and morality if there is no global recognition? One will be victimized the other will be benefiting the victimization of the others. How you respond to this fact? It is in reality right now, which peoples are being hacked by their throat through this means. Thank you.
I think
If you like, yeah.
Please do. Okay.
I think that our president, Dr. Arif Alvi, in the morning said that this is a neo-colonialism. He used this word. I think this is the same everywhere, not only in agriculture, but all multinational companies. And so it's not, instead of coming and invading countries, you have economic policies that strangle countries. So this is going on, but we cannot do much about this. What we can do is that minimize this by having indigenous research and having our own solutions, or working together as we are doing today. So multinationals are there to make money, and unfortunately, the world is controlled in a way that you have what we call as trade barriers. So if you impose any trade barrier, there will be complaints. So this is a more like a political issue. But I think that what we can do is actually take some efforts and use science to come up with our own solution, instead of just waiting for the solution, and accept what is good for the country, or maybe and what is not good for the country. But the policies, international policies are like this, that WTO regime, it's very difficult to control any trade, to have any trade barriers. So this is our reality.
Okay. Normally, yes. Yeah.
Question is from Dr. Asim. First of all, thank you for your very nice presentation. Actually, as you know, that contamination is one of the very limiting factor in tissue culture. I have been working extensively on plant tissue culture for the past 10 years, and this is something we are not able to control.
Can you speak up a little bit?
I just want to have your 2 cents on the innovations in contamination.
Microphone is not working, I guess.
Yeah.
So he's asking about the contamination problem.
Yeah. So may I ask which crop you worked? First word is important.
Actually, we are not working directly on plants. We are working crops. Sorry, we are working on medicinal plants.
Okay.
But the problem is that contamination, usually, as you talked about shortcuts in plant tissue culture, we have been using shortcuts like using antibiotics, but you know that antibiotics have their limitations and their problems too. So could you comment on the fact that using antibiotics is a good strategy regarding tissue culture or not? Or what could be more innovations to control contamination in plant tissue culture to get more and more benefit from plant tissue cultures?
You know that if the contamination is exogenic, it's very easy to control. If it's endogenic, that is a big challenge, especially in the medicinal plants. And if you check, if you focus on that point, normally there is no contamination in the culture media. Whenever you cut the plant for a subculture, you can find the contamination there. Is it right? And it is due to the endogenic. And normally this type is the bacterial contamination, not the fungal contamination. Fungal contamination started within a few days. It will not remain within the plant. So right now, the antibiotics is the best right now. But you can use this nanoparticle like silver nanoparticle. That's a good opportunity. You cannot use this antibiotic, and you can overcome the issue by using this nanoparticle, but the concentration is very important. And one thing, you can use these nanoparticles before autoclave. There is no issue, okay? And the dose is important.
So sorry, I didn't get the point. So we have to add before autoclave or after.
These
autoclave?
nanoparticles are basically synthetic and they can tolerate up to 800 degrees Celsius temperature. So you can directly use prior to the sterilization in autoclave. You can use before autoclave.
Before.
It's silver nitrate, but silver nanoparticles are much smaller, so they can enter within the cell culture cells and they can control it.
Next question.
Okay, Dr. Zain is here from Agriculture University, Faisalabad. I have very specific questions and one journal along with very short comments. First of all, my specific question is that regarding the development of biotechnological products like biopesticides, bioinsecticides, are there any sustainable solutions through the innovations or interventions of biotechnological techniques to overcome the resistance development in crop productions, particularly in the insects? I want to ask from the Sir Mansoor.
I think that, the answer is that, for example, so resistance is something which is expected very much, it's just the timeline that, so what you do is that you work on more genes, identify more genes. It's just like, antibiotic resistance, that you have resistance against some antibiotic and then you switch to another group of antibiotic. So same way, if you have resistance to one gene, you can quickly adopt to another gene or combination of genes. For example, in case of BT, we are now moving towards 2 or 3 genes to have a better control. But also there are ways to delay this, use of refugia and other technologies to, we can delay it, but we cannot overcome resistance is something which is going to happen anyway.
Okay, second part is that it's a general comments. In order to feed our ever-growing population of the world, we have to rely on biotechnological interventions. Otherwise, we are unable to meet the challenges of food security. Although, along with that, in developing countries, along with Pakistan, I want to know what are the risks and constraints and the challenges which we are unable to satisfy the commercialization authority so that different biotech products are commercialized in Pakistan. It is not a problem in Pakistan, but almost in all of the Europes, including the UK and the underdeveloping countries. Either we don't have sufficient data to convince them to support our products. Sir, what do your comments over this one?
Would you like to? Okay, I think that the barriers are sometimes it's a mindset that you don't want to listen and you don't want to do something, which again pointed out by the president in the morning that we have a barrier to technology. So this is something that especially, but unfortunately the role which is played by denial of the technology by Europe is actually a very big concern, because we are caught between the American model and European model. But fortunately, for some of the emerging technologies like genomics and genome editing, there is much less problem. UK is very much moving fast into commercializing this genome edited crop. So I think that the situation will improve in future, because as I said that a lot of traits like quality, yield, they will come through editing of some of the genes which are negative regulators. So we hope for the best.
Here is Dr. Muhammad Reem from Khwaja Farid University of Engineering and Information Technology, Rahim Yar Khan. So my question is to Professor Dr. Shahid. How much GMO seed we import from other countries?
There's no import of GMO seed. The only thing is that we are importing GMO soybean, and this is not for seed purpose. This is as a grain. And right, after import in Port Qasim, this is processed mostly in solvent extraction plant, which are located in Karachi. So I mean, if there's a minimum, risks, because, those are not imported as seed, but as a grain.
My second question is to Professor Dr. Shaheen. Madam, how much sugar content we have improved for sugarcane?
In our line?
No, overall in our country.
Overall is the sugar content, you can say the bricks content lies in 19% or 20%, but in these lines we found a 22%. So even if you find a 1% increase, is a lot of increase in the sugar content.
Thank you, madam. My last question is to Professor Dr. Turki. Sir, I want to know about how we can quantify before the experiment, how we can quantify the data. You have told 4 steps. The last step is the quantification of the data. So can you explain?
Yes. This is something new in machine learning. It depends on your experiment actually. For example, if you say the quantification means you can distribute your data into different subgroups. It's called the clustering method. So you can use clustering method and you can distribute your data into different clusters. But before that, the input is very important in that experiments. Yes. That is very important for that.
Thank you, sir.
Probably we can take 1 or 2 more questions so that
last 1 or 2 questions.
Yes, I think that's from this side. Students, ladies.
Yes.
I have a question from Dr. Asim. So it's regarding tissue culture. So what are your insights on morphogenic regulators for the enhancement of regeneration? Have you tried that?
So I can't understand your question. Can you repeat, please?
What are your insights on morphogenic regulators, like growth regulating factors that has been reported in wheat and baby boom and gene from Wushell family?
He's asking about the use of baby boom. This is actually a technology, a baby boom gene.
Yes.
Morphogenic regulators.
Yes.
To enhance
So
the regeneration.
I didn't work on that aspect right now, but AB, I think, worked on that. I was involved in the thesis, I think. So I have no information about now on that aspect.
So the second question is from Dr. Emre from the previous session. So you have told that we can sequence a whole genome sequence the orphan crops. So rather than going for whole genome sequencing, can we do the targeted sequencing from the from the data or candidate genes that we already have? So that is very expensive, whole genome sequencing. So even single SNPs can influence the whole trait. So what are your thoughts about that?
So there was a hand raised. So answer is yes. Okay.
Okay, last 2 questions from there. So then we will close the session.
Assalamu alaikum. I'm Dr. Nishaman from GIBG. I just want to know there had already been a lot of discussion about GMOs, and I was just confused. I usually get confused when I come across these things that genetically engineered crops usually have a lot of issues. And now people are more talking about genome editing. So actually, what is the difference between a genetically engineered crop and a genomically edited crop? Are there different biosafety regulations? And if it is more acceptable, maybe we should be making waves in that particular area.
Yeah, I think that there's a very clear policy. If there's no foreign gene inserted while in genome editing, so it is considered as non-GMO. We are following the same policy in Pakistan. The data is the paper is just accepted in Journal Gene. And I think that we are following the policy of some of the other nations like Japan and Australia that if there is no foreign gene, it should be considered as non-GMO.
So it is internationally acceptable also.
Yeah, many countries accept them as non-GMO because it is equivalent to creating some mutation. Earlier we were using radiation chemicals. Now we are using editing CRISPR to do editing.
Thank you.
The last question, I think.
Assalamualaikum, I'm Ume Kulsum. I'm a PhD research student at KIPG. I was wondering, just like we can utilize different kinds of light in order to enhance plant tissue culturing, are there any studies where we can use different frequencies or sound waves in order to enhance plant production?
So you are talking about the mass production, is it?
Yes.
Yeah, of which crop? That is important.
Are there like at the basic level? At the basic level, are these kind of techniques being utilized in the field?
Yes, now tissue culture started from the lab, now converted into the commercial propagation, and now it's going towards the hobby. Okay. If you check the groups on the social media, you can find groups on the plant tissue culture. These are the non-professionals. They are just develop their own protocols, finding out from the YouTube, and now they are selling these plants online to all over the world.
Okay.
So mass production is possible for every crop, but it depends on the plants. If you are going for this ornamental plants, that's a good. Some root stocks, $1 million company, but not prevailing in Pakistan. So it's possible for every plant to go for the mass propagation. It's very easy.
Okay, thank you.
I think one question last. Aftab just raised the hand, so I think that, you know.
Sir, I have a question directly related to you that I just want to ask, are you not member of regulatory authority controlling GMO in Pakistan, GMO release in Pakistan?
Yeah, I mean, we are part of the what we call as technical advisory committee.
Okay.
So, so some of the, I mean, for example, you know, say we can discuss the case of GM maize. It was recommended by the technical subcommittee, but not approved by National Biosafety Committee.
Yes, because sir, India has allowed genome-edited crops as well, and EU is expected to revise its regulation.
I think that, you know, so far we have not received any genome-edited crop commercialization in Pakistan. We are sure that, you know, if such cases are, you know, we receive that tech, we will recommend that, because if there's no GM, no problem.
Thank you, sir.
This is our stated policy, and this is going to be published very soon.
Thank you, sir.
Okay, we should close the session.
Yeah, thank you very much to all speakers, and it was been a wonderful session and a lot of good questions. So I hope that, you know, we will continue have discussion on tea, dinner, and you know, while just talking or walking. So continue and have a thought process. I have a lot of questions to Dr. Asim, which I will ask separately. Thank you very much.